All right, well, welcome back to Glycolysis 101. So we introduced this overview last time, and what we said is that glycolysis is a pathway for taking sugar, and the specific sugar we're talking about here is glucose, but other sugars can feed into glycolysis as well. They just have to get converted into glucose or into something that's a little further down the line. And a couple of our high-energy carriers in the cell, ATPs, are going to get invested to kick this glucose up to a higher level of free energy, but that will then position it to release energy in other reactions down the road. There's a couple of important points about glycolysis I want to make before we go on. One of them is that the reactions occur in the cytosol. They don't occur in mitochondria or peroxisomes or ER or any kind of an organelle that's restricted to a eukaryotic cell. Second thing is that it occurs without the use of oxygen. Now, we're going to look later on at a portion of catabolism that does depend on oxygen. And so cells that live in places where there isn't any oxygen are kind of left out of that portion of metabolism. But glycolysis can happen in any type of cell. And so it's pretty much universal, whether we're talking about bacteria or yeast or mushrooms or oak trees or human beings or sperm whales. Everybody's carrying out glycolysis. This is kind of the basis of catabolism. And we can break glycolysis down into three parts. The first part is going to be where we invest that energy from ATP to kick up the free energy. Second part is going to be where we split this 6-carbon sugar into 2 3-carbon molecules. And then the third part is where we're going to get back activated carriers in the form of the electron carrier NADH and now ATP. We're going to get back more than we put in. And you say, how is that possible? You know, first law of thermodynamics says you can't do that. Well, remember that all of those covalent bonds in the glucose have energy to give up. And so it isn't just the energy that came from the ATP that's being recovered. It's energy that was stored in those covalent bonds between carbon and carbon, between carbon and oxygen, between oxygen and hydrogen. That bond energy is going to be extracted as well. And that's what allows us to end up with a net gain of activated carriers when we catabolize glucose. So let's look at the first part, the energy investment stage. That's the first 3 reactions out of 10 that occur in glycolysis. And I think I said this last time, but I'll reiterate it again. I'm not expecting you to remember the structures. I'm not expecting you to remember the names of the intermediates. I'm not expecting you to remember the names of the enzymes. I'm not expecting you to be able to draw the reaction mechanisms. I'm going to show you all that stuff because I don't want it to be a black box to you. I want you to be able to see what's going on. But what you're going to be responsible for memorizing, that is, what I am going to test your knowledge on, is do you get the big picture. So just kind of relax, take a deep breath, don't get too hyped about structures, and let's see what happens. So the first thing that happens when glucose gets into a cell, and how's glucose going to get into a cell? Is it just going to diffuse through the membrane? What's it going to do? It's going to what? Okay, it's going to use proteins where? On the cell membrane, correct. And there's more than one type of transport protein that brings glucose into cells. Some of them are passive transporters. They depend on there being a gradient of glucose concentration across the membrane, higher outside the cell, lower inside. Some of them are active transporters. They're secondary active transporters, meaning that they use potential energy of an ion gradient that was created by some primary active transporter to then bring the glucose into the cell. And so depending on what tissue we're talking about, what kind of cell we're talking about, what environment it's in, there's different ways glucose can get in. Yes? Well, you can endocytose stuff that has glucose as part of its structure, And you can endocytose whole organisms if you're a phagocyte. They got plenty of sugar in them too. But that's not specifically targeted at bringing glucose in. Endocytosis, and when we talk about it, we'll talk about the different forms. It is not the primary mechanism for bringing small molecules into cells. Now, there is a form of endocytosis called penocytosis that just pretty much grabs fluid from outside the cell, and whatever's in it comes into the cell that way. But that's not your primary way of bringing it in. But yeah, you can get some in that way. But once the glucose is in the cell, something's going to happen to it almost immediately. And what that is a reaction with ATP to put a phosphate on the glucose. And that phosphate is going to be added right here at the hydroxyl group that's highlighted in blue. And throughout this, they're always going to highlight the portion of the molecule that's involved in the reaction. So that's what the little blue boxes are all about. So we're going to split our ATP molecule. And everybody knows that when you split ATP, you release free energy. Okay, so we're going to couple a free energy releasing reaction to some other reaction here. That splitting is carried out by the enzyme hexokinase, and hexokinase is going to transfer the phosphate that came from the ATP onto this carbon-6 hydroxyl group here, creating a phosphoryl bond. Now, a phosphoryl bond is not considered a high-energy bond, which kind of makes sense. That is, we couldn't sacrifice one that is a high-energy bond and create another one that's just as high energy. First law says we can't do that, or really the first and the second law say that. So we're creating a bond between a phosphate and an oxygen here, but it's not got enough energy stored in it yet to do any work for us. But it does accomplish something. First of all, it sets us up for a later intermediate that is going to have enough energy to transfer phosphate to ADP and make ATP. Second thing it does is it changes our glucose molecule. Okay, originally we had a molecule that was polar, but it didn't have any charge in it. Now we've put a phosphate on it. Now we always abbreviate phosphate as just our little yellow circle with a PI in it. Just means inorganic phosphate. That's fine. But remember that a phosphate ion is negatively charged, has two negative charges on it. And therefore, is there much chance that our now phosphorylated glucose is going to get back out of the cell? No. It certainly cannot get across the membrane all on its own. And the transporters that brought it in, whether they were secondary active transporters or they were passive transporters, they're made to bind glucose, not glucose 6-phosphate. So therefore, once that glucose is phosphorylated, it's locked inside. Furthermore, if we're looking at passive transport, that is, we're trying to maintain a gradient of high glucose outside to low glucose inside, then when we convert glucose to glucose 6-phosphate, it isn't glucose anymore. Therefore, that gradient still exists. So this first step accomplishes two things. It primes the pump, if you will. It starts the process of creating a higher energy version of the carbohydrate molecule. And second of all, it traps the glucose inside the cell. Step two in the energy investment process is going to be an isomerization, a rearrangement of the structure. And the cell is going to take advantage of the fact that 6 carbon sugars can exist in 2 different ring conformations. We already saw the one. Now we're going to look at a different one. This is a 5-membered ring instead of a 6-membered ring. Still 6 carbons. 1, 2, 3, 4, 5, 6. We didn't lose any carbons anywhere here. We just moved one of them outside the ring, whereas before it was part of the ring. And this reaction is carried out by an enzyme called phosphoglucose isomerase. Not a real big free energy difference between the product of this reaction the fructose 6-phosphate and then the glucose 6-phosphate that was the substrate for the reaction. So not a lot of energetic push needed here. But this rearrangement is possible because sugars, monosaccharides, have this unusual capability of reversibly flipping from a ring structure, like this one or this one, to a linear structure. And that happens because of events here at this bond, not the one that's highlighted here, but this one here. And glucose is an aldose, meaning that in its linear form it has an aldehyde group, C double bond O, with a hydrogen here. And once the sugar has spontaneously converted from its six-membered ring form into the linear aldose form here, it can either go back the way it was, or it could be converted to a five-membered ring. And that's what the phosphoglucose isomerase does. It grabs the linear form of the aldose here, and then it simply now creates a new bond. Instead of carbon 1 attacking carbon 5 here to make a six-membered ring, it attacks a different carbon here, and that's going to leave us now with one of the carbons outside of the ring. I should mention, you have to isomerize from the aldose form to the ketose form here. I kind of left that out. Can't leave that out. And so now carbon 2 is going to attack carbon 5 here, and we're going to leave carbon 1 here outside the ring. This doesn't change the energy level of the molecule any. Why do you think there might be a good reason biochemically to do this? Why are we changing our six-membered ring to a five-membered ring? Look for the big pink words at the bottom of the slide. Well, the ring is five-membered. You can say it's smaller, but that's not the word that's on the screen. What's the word on the screen? Symmetrical. What does symmetrical mean? What's identical? Both sides. Both sides. That is, we're creating a molecule that you could draw a line through the middle of, and the two sides would be pretty much the same. Now, they're not exactly the same, because in the five-membered ring here, we've got a carbon with a hydroxyl group here, and we got one with a phosphoryl group over here, so they're not identical. I saw somebody with their hand up back there. Do you have a question? Or not really. That's not really what we're talking about here. Yeah. You're getting ahead, but you're right. Remember that we're going to split this molecule in two, and we're going to make two products that are identical, the pyruvates he's talking about. And to do that, we need to first convert our carbon skeleton into a structure that's going to allow us to make two identical three-carbon molecules, or as we'll see, two almost identical three-carbon molecules. So that's what we're setting up here. We're creating a more symmetrical carbon skeleton, one that we can split in two and get back products that are essentially identical to each other. But we've got to do something else first. We've got to balance the other side. We've got to make carbon 1 be in a phosphoryl linkage 2, like carbon 6 is. And that'll really make our molecule symmetrical. So this is where our second ATP investment comes in. And the enzyme that carries out this reaction, phosphofructokinase, is the primary regulatory enzyme for glycolysis. And the reason it is that the free energy change in this reaction is very high. It's very high. And in order to run this reaction the other way, you would have to put so much free energy in that essentially under normal cellular conditions you couldn't get the reaction to go. And so up until this point, you can, under the right circumstances, drive the pathway forward or backwards. Okay, and that's going to be significant later when we talk about gluconeogenesis. But this reaction has such a high negative free energy change when it runs this way that means it has an equally high positive free energy change going this way, meaning you'd have to add so much energy to get the reaction to go this way that it just doesn't happen. And when you look at how biochemical pathways are regulated, you often find out that that's true. A step with the biggest favorable free energy change is going to be the one that is the main regulatory point for the pathway, and that's true of glycolysis. So that makes this particular step important. It's the committed step of glycolysis. Once you've gone past this, once you've converted fructose 6-phosphate into fructose 1,6-bisphosphate, you're not going back. So now we have a molecule that is symmetrical. I can draw a line right down the middle of this, boom, and I've got two halves that are identical to each other. 3 carbons and a phosphate group. So now we are set up for the next phase of glycolysis, splitting the sugar ring in two. And that's what's going to happen. The enzyme that carries this out is called aldolase, because it's splitting an aldol. Aldol is a molecule that has both an aldehyde part and a alcohol part. That's the ol, so aldol. And this guy is going to be split in two. It's going to, again, interconvert between the ring and the linear form, and these two carbons, 3 and 4, are going to be the site at which the enzyme breaks the bond. And that's going to yield two products. One is glyceraldehyde 3-phosphate. Okay, so that's going to have these 3 carbons here. And the other one is dihydroxyacetone phosphate. That's going to have these 3 carbons here. Now we want to end up with 2 identical molecules. These molecules are not identical. They're similar. They both got 3 carbons. They both got a phosphate attached. One of them is an aldehyde, though. Here's our aldehyde group. One of them is a ketone. Here's our keto group here. So if we want to keep going, we got to find a way to end up with 2 identical products. Happily, interconverting aldehydes and ketones is not that hard. And that's what's going to happen. Another enzyme that carries out an isomerization reaction is going to grab the dihydroxyacetone phosphate, the ketone of our pair of 3-carbon molecules here. And it's going to convert that keto group to an aldehyde group. Now we're going to get into some resonance structures that are formed in the intermediate, and we're going to interconvert now our aldehyde and ketone groups. And when we do that, what we end up with is glyceraldehyde 3-phosphate, exactly the same as the other product that we made in the last reaction. So now, where we had 1 glucose molecule originally, we have 2 glyceraldehyde 3-phosphates. So now we're set up for the third phase of glycolysis, where we're going to get energy back. Now, we're not ready yet. I told you these phosphoryl groups, where you have basically a hydroxyl converted into a phosphoryl linkage here, don't have a lot of energy stored in them. That is, if you split that bond, you'll release some energy, but it's not enough energy to make an ATP. Remember, when you split the phosphate off an ATP, you release 7.3 kilocalories per mole. And if you want to make an ATP, you've got to be able to put in more than that, because you're always going to lose some as heat. So you've got to have some reaction that yields more than that. And splitting this phosphate off a glyceraldehyde 3-phosphate isn't going to do it. Not enough energy stored there. So we've got to change things. So first thing we're going to do is carry out a redox reaction. Everybody knows what redox reactions. What's going to get moved? Electrons. Electrons are going to get moved here. And so here's what's going to happen. There is an enzyme called glyceraldehyde 3-phosphate dehydrogenase. This uses NAD+ as a cofactor. And when you've got an organic molecule that's a cofactor, anybody remember what we called it? It's a diffusible organic molecule that's a cofactor. We had a word for that. Coenzyme. Everybody hear that? Coenzyme. So NAD is going to be a coenzyme for this particular reaction here. And so what's going to occur is that we are going to take this aldehyde group here and we're going to oxidize it to a carboxyl group. Remember, in our hierarchy of carbon oxidation, aldehydes were more reduced than acids were. And so we are going to remove electrons from an aldehyde, transfer them to NAD+, and be left with a carboxyl group that is going to pick up a new phosphate. Where's the phosphate come from? Well, it's going to come from just inorganic phosphate, PO4, that's floating around in the cytosol. Okay, there's always plenty of inorganic phosphate around. So in the reaction mediated by this enzyme, we have now oxidized this particular carbon one level, and we've created a new bond to a phosphoryl group here. But unlike the bond that exists down here, where we have a carbon at the level of an alcohol that's coupled to our phosphate, here we have a carbon that's at the level of an acid coupled to our phosphate. What that means is, for organic chemistry fans, there can be some resonance energy here. That means that bond is more unstable. The phosphate is going to be a better leaving group, and you're going to release more energy when you split that bond than when you split this kind of bond. So we now have a phosphoanhydride bond, and splitting that would give you enough energy to transfer your phosphate to ADP and make ATP. You know, we kind of think of ATP as being the pinnacle of our energy molecules, right? Because that's all we ever talk about. But you're going to see here, there are molecules, phosphorylated molecules, that have more free energy of hydrolysis in them than ATP does. And if you've got one of those guys, you can transfer the phosphate from that molecule to ADP, and you can make ATP. It's energetically favorable. So now we have something called 1,3-bisphosphoglycerate. We have a high-energy phosphoanhydride bond to our first carbon, but we still got a low-energy bond down here. So we're going to capture the energy stored in this phosphoanhydride bond right now. And that's done using an enzyme called phosphoglycerate kinase. Now, you're going to hear repeatedly in this course that a kinase is an enzyme that takes ATP and transfers a phosphate from it to something else. And that's actually how it's defined. Here, though, we're talking about using a kinase to take a phosphate from something else and transfer it to ADP. Why do we call it a kinase? Well, remember that all enzymatic reactions are, in theory, reversible. It depends on the conditions. It depends on the free energies of the reactants and the products in a given scheme. Are you running this way or are you running that way? And of course, it depends on other things, temperature, pH, so on and so forth. In this particular case, 1,3-bisphosphoglycerate has a higher free energy of hydrolysis for this bond here than the free energy of hydrolysis for splitting a phosphate off of ATP to make ADP. That means it's actually energetically favorable to run the reaction backwards from the way the name of the enzyme would make you think it goes, and to make ATP instead of splitting ATP. And that's exactly what happens. And so phosphoglycerate kinase is going to split this phosphoanhydride bond here, transfer the phosphate to adenine diphosphate to make adenosine triphosphate here. And so now we've made an ATP molecule. And remember, we had two 1,3-bis phosphoglycerates, because we started with a 6-carbon molecule and then we made two of these guys. So we're getting two ATPs out of the leftovers of our glucose molecules. Where does that leave us energy-wise now? We still behind? Are we ahead? Where are we? In terms of ATP, we're equal. What about in terms of overall energy captured? Think about what we made in this reaction here. We're actually ahead because we captured some energy in our reduced electron carrier, NADH. So yeah, we've canceled our ATP debt now, and we've made a reduced electron carrier. So we're actually energetically ahead now. We're starting to win the game of extracting energy from glucose. This reaction, in which you transfer a phosphate group from an organic intermediate to ADP to make ATP, is called substrate-level phosphorylation. And substrate-level phosphorylation occurs not just in glycolysis, but in other types of biochemical pathways as well. I'm going to contrast that with oxidative phosphorylation that we'll talk about later, in which you use electron transport to create a potential energy source that's going to be used to drive ATP synthesis. That's a different mechanism altogether. But substrate-level phosphorylation means that you take an organic substrate, you transfer a phosphate from it to ADP, and you make ATP that way. So now we have depleted one of our high-energy groups here from our 3-carbon skeleton. But we still got a phosphate here. And we need to set that guy up so that he is in a high-energy phosphate bond that can be split and used to transfer the phosphate to ADP to make ATP. And the first step in doing that is changing the position of the phosphoryl group in the 3-carbon intermediate. So we had 3-phosphoglycerate. We're going to move this phosphate to the 2-carbon here. And the enzyme that does it is called phosphoglycerate mutase. Very interesting enzyme. We won't get into the mechanism here. If you took 342, you heard about this. If you're going to take it, you'll hear about it. When you simply look at this, you say, okay, the enzyme somehow breaks this bond, it grabs the phosphate, and it moves it over here, and it sticks it on there, right? End of story. That's not actually what happens. What happens is that the enzyme comes in with a phosphate already attached to, I believe it's a serine in its active site. Then it binds the substrate, and it sticks its phosphate on carbon 2. So you have an intermediate that has 2 phosphates on it temporarily. But then the enzyme purloins this phosphate here to rephosphorylate its serine, and then it releases the product that it's made, 2-phosphoglycerate. So there's actually a hidden phosphate in there that you don't know about when you see the reaction drawn this way. Really interesting enzyme. But now we have repositioned our phosphate and we've set it up to be converted into a high-energy linkage. And we do this by using an enzyme called enolase to kick water out of the molecule. And when we do that, we're going to convert this bond here from a single bond to a double bond. Now, once again, we're setting up a potential for resonance structure. That is, you can flip back and forth here between double bond here and double bond here. And that causes this bond to become unstable. That is, this guy is going to be a high-energy molecule. If you break this bond, allow phosphate to leave the molecule, you will actually stabilize the remainder of this molecule and you release a lot of free energy. So this compound is called phosphoenolpyruvate, and phosphoenolpyruvate is one of the few compounds around in our biochemical systems that has more free energy stored in it than ATP does. That means that you can actually transfer a phosphate from that to ADP to make ATP, and it's energetically favorable. And so that's what's going to happen in step 10 of glycolysis. You're going to take that phosphoenol pyruvate, you're going to split that unstable linkage here, you're going to transfer the phosphate to ADP to make ATP, and that's going to leave you with pyruvate. And so now we have completed the process of glycolysis. We are now ahead by 2 ATPs because we canceled our debt earlier, and now we just bought 2 more. Why do we have 2 more? Not just 1 more? Because you had 2 3-carbon molecules from your 6-carbon starting material. That's exactly right. And so we've now gotten ahead by 2 ATPs, and we got ourselves 2 NADHs earlier when we oxidized our 3-carbon molecule. So now we have carried out the very first stage of respiration. We have begun the process of extracting chemical energy from a sugar. And a sugar you can think of as being a polyalcohol, has a lot of hydroxyl groups on it. And we've created something now that is a keto acid. Here's our ketone group here. Here's our carboxyl group that makes it an acid here. So we have produced more oxidized product than we started with. And along the way, we have captured some energy in the form of the activated carriers ATP, of which we've gained 2, and NADH, we've also gained 2 here. So that's a way to make some ATP, and it's a way to make some activated electron carrier here. And you know what? For some organisms, that's all they can do. They can do glycolysis, and they can't do anything else. However, there's still a lot of energy stored in this pyruvate. So if you aren't able to further oxidize that molecule, you're leaving a lot of energy on the table. But sometimes that's what happens. One reason that it might happen is that to further oxidize this molecule, you need to have some kind of an electron transfer system with a terminal electron acceptor, and that terminal electron acceptor is often oxygen gas, O2. So one reason you might not be able to oxidize your pyruvate any further is there's no O2 around. If you live in an environment where there's no O2, and believe me, there's plenty of them on Earth, then you're stuck with glycolysis. There's another reason, besides availability of O2, that some organisms don't oxidize pyruvate any further. Can anybody think of what it might be? Yes. They don't have the enzymes, meaning they don't have the genes. They simply aren't capable of producing the proteins that are necessary to carry out additional oxidation of pyruvate and get more ATP. So they stop there. And that's fine. They can't do all the same things that an organism that can extract more energy can, but they can still make a living. So what these organisms do is they ferment. Now, fermentation is really just glycolysis, but with a catch. Remember that in glycolysis, we carried out a redox reaction and we made some NADH. To keep glycolysis going, there always has to be a supply of the inactive form of the NAD coenzyme, NAD+. If you keep running glycolysis and running glycolysis and running glycolysis and running glycolysis, what's eventually going to happen? What's going to run out? NAD+. You're going to convert it all into NADH. And when that happens, you got a problem because the equilibrium says we're not going to go back the other way. And so without any coenzyme that's able to accept electrons, glycolysis can't run anymore. Without running glycolysis, you got no way to make ATP. Without ATP, you got no energy for reproduction. And as I always say, you reached equilibrium. you're a dead cell. So there has to be some way to get back our oxidized coenzyme, and that's what fermentation is about. Fermentation is going to be glycolysis, and then one or two additional steps that are going to get you back your oxidized coenzyme. There are two major forms of fermentation I want you to know about. One is lactate fermentation, which occurs in many types of microorganisms and in your muscle cells. The other is alcoholic fermentation, which can occur in multiple organisms, but we mostly associate it with yeast, which are single-celled eukaryotic organisms. And both of these are just two twists on the same idea, that you've got to somehow get back your NAD+. So in lactate fermentation, what you do is take pyruvate, an alpha-keto acid, and using an enzyme called lactate dehydrogenase, you reduce this pyruvate. Remember, we've been oxidizing things all along here to get to the level of pyruvate. Now we're going to go back a step. We're going to reduce our alpha-keto acid to an alpha-hydroxy acid. So here's the more oxidized form, carbonyl group. Now we're going to reduce it to a hydroxyl group. And this product is lactic acid or lactate. And this particular enzyme, lactate dehydrogenase, requires NAD as a diffusible cofactor. But this time, what's going to happen is that you're going to transfer the electron to NAD, or excuse me, to the organic substrate here from NADH to regenerate the inactive form of the coenzyme, NAD+, and give you this more reduced alpha-hydroxy acid. So that's one strategy for doing it. In alcoholic fermentation, you first decarboxylate the pyruvate. That is, you break this bond here. So that's why your beer has fizzy bubbles in it. That's why if you make wine, you have to have a device there to let the CO2 out during the process of fermentation. It comes from the decarboxylation of pyruvate. And then an enzyme called alcohol dehydrogenase is going to do the same thing that lactate dehydrogenase did over here. Using NADH as a cofactor, it's going to reduce the product, the two-carbon molecule left here, to ethanol, CH3, CH2OH, and it's going to then regenerate your inactive NAD coenzyme over here that can then go back and be used in glycolysis. And there are lots and lots and lots of other types of fermentations that exist in nature. And you can make all kinds of different organic compounds from them. You can make alcohols and aldehydes. And in fact, a lot of the commercial production of solvents that you use in chemistry class is done using fermentation reactions. You just have to know the right microorganism that's got the right set of enzymes and produces the product that you want. Yes? You could consider it that, but it's really not. You're not making a more complex molecule. You're simply carrying out a redox reaction. Okay? So when we talk about anabolic reactions, we're usually talking about joining small things together to make bigger things. So no, we don't normally consider it that. But you're right in the sense that in general we think of catabolism as being an oxidative series of reactions and anabolism being a reductive series. And here we are going back one step in the reductive direction, but we're not going to do anything with this end product. That's the end of the road. It's released as waste by the organism. Now, it's not necessarily waste to us. You know, those of us who like our beer and wine and whatever, we're very happy about the fermentation producing alcohol. If we like to eat dairy products, if we like yogurt, if we like cheese, if we like coffee, if we like chocolate, all of those are products of fermentation reactions. And the organic end products of the reductive reaction that occurs are what gives these foods we like to eat their flavor. And so we exploit this process for our own gain here. But from the organism's point of view, all it's trying to do is keep its glycolytic pathway operating by making sure that it completes this cycle and it converts its reduced NAD coenzyme back to the oxidized form. One thing that I want to point out, because you guys will run into this, especially you guys that are interested in healthcare and you go on to medical school or osteopath school or whatever it might be. In a lot of textbooks, for reasons known only to whoever, the term anaerobic respiration is applied to fermentation. That makes microbiologists and biochemists pull their hair out. Respiration has a very specific meaning in biochemistry, and that is that you are using electron transport to drive the production of ATP, and that you are completely oxidizing, or at least almost completely oxidizing, your fuel molecules. Neither of those things happens in fermentation. You do not further oxidize the pyruvate that's made in glycolysis, and you most certainly do not use electron transport in any way. So if you, in a textbook, whether it's a medical school textbook or any other, see the words anaerobic respiration applied to fermentation, take a pencil and cross it out. That's not right. When you're talking about fermentation, you're talking about fermentation. The reason I don't like people to misuse that, it wouldn't matter if there weren't such a thing in nature as anaerobic respiration, but there is. There is, and we'll talk about anaerobic respiration, but it's a completely different animal than fermentation. So here's the things to know about glycolysis and fermentation. It's a cytosolic method for partially oxidizing glucose to pyruvate. And since you're only partially oxidizing your fuel molecule, you're only getting some of the energy out of it that it has to give. Glycolysis has 3 stages to it. You should know what they are: energy investment, splitting the molecule in 2, and then energy recovery. You're getting back the energy that you invested plus some more. You should know what the products of each of these phases are. The first one, it's the fructose 1,6-bisphosphate. The cleavage phase is giving you glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, but you're going to convert the latter to glyceraldehyde 3-phosphate, so you can essentially say you're getting 2 glyceraldehyde 3-phosphates. And then the third phase, the products are going to be your NADH, your ATPs that you're gaining, and the pyruvate. So make sure you know those things. How much ATP do you put in versus how much do you get out? Well, you put in 2, you get out 4, so you gain 2. Is oxygen used in this? Did you guys see oxygen gas anywhere in there? Nope. And there's something else I'll caution you about. Because of that, you'll sometimes see glycolysis referred to as an anaerobic process. And it is. However, it's not unusual for people to hear that and then presume that means that this process can only take place if there's no air, if there's no oxygen around. And that's not true at all. It's anaerobic because it doesn't use oxygen. It's not anaerobic because it can't tolerate oxygen. Doesn't matter. Oxygen just simply isn't utilized in this pathway. Is ATP the only energy-rich molecule you make in glycolysis? What's the other one? NADH, right. Don't forget about the NADH. Glycolysis made ATP through substrate-level phosphorylation. What did that mean? What did substrate-level phosphorylation mean? From what? Well, you're getting at it. It's actually a little simpler than that. It's adding a phosphate to ADP from what? From an organic intermediate, right. That's substrate level phosphorylation. So know what that means. Which is the reaction in glycolysis that was the primary point for regulation? It's that third one, okay? It's that third step where you added the second phosphate and you made fructose 1,6-bisphosphate. That's the one that has the biggest negative free energy change and is not going to want to go back the other way in cells. And we'll see later how you, or some ways I should say, you regulate that particular reaction. What occurs during fermentation? Fermentation is just taking glycolysis and putting one redox step at the end of it that allows you to regenerate your oxidized NAD coenzyme. And why isn't it equal to anaerobic respiration? Because to be respiration, you got to have electron transport and you got to be completely oxidizing your fuel. And you don't do that in fermentation. And then why are these reactions so important? If you didn't regenerate your NAD+, what couldn't happen? Glycolysis. You couldn't keep carrying out glycolysis if you didn't regenerate your oxidized NAD coenzyme. That's why it's important. All right, so we're going to wave goodbye now to the microorganisms that don't have the enzyme systems to carry out further oxidation of their pyruvate, or they simply live in a place where the oxygen that they're going to need to do that isn't available to them. And incidentally, there are a lot of microorganisms that can swing both ways. We call them facultative anaerobes. And in what's usually considered their normal home, there's no oxygen around and they carry out fermentation. But if they find themselves in an environment where there is oxygen available, they have all the tools to go all the way to full oxidation. Good example are the bacteria that live in your gut. Now, there are some of them that are obligate anaerobes. They can't survive outside your gut where there's oxygen. But there are guys like E. coli and Salmonella. And most of us don't have Salmonella in our guts, but if you're a chicken, you do. And those organisms, when they find themselves outside the environment of the colon, which is their normal habitat, and there's oxygen available, they can switch on their system for complete oxidation of fuels, that is aerobic respiration. And they can grow like wildfire, and that's why sewage treatment works. It's also why when raw sewage gets places it isn't supposed to be, you can have problems. We're going to focus now on the eukaryotic cells. These are the guys that have the tools to carry out complete oxidation of their pyruvate. To do that, they've got to get the pyruvate into one of the organelles, and that's the mitochondrion. And so the first obligate step beyond glycolysis is going to be transporting pyruvate from the cytosol into the interior of the mitochondrion. Now, the mitochondrion, you remember, has two membranes. It has an outer membrane, and then it has a very heavily enfolded inner membrane. The inner membrane is where a lot of the business of respiration is going to take place. The rest of it's going to happen in the matrix, which is kind of the cytosol of a mitochondrion. It's the fluid, water-based environment that exists in the interior of the mitochondrion within the inner membrane and contains all the enzymes and the other molecules that are necessary for you to complete oxidation of your fuel molecules. The outer membrane has so-called porin proteins in it. Porins are kind of like channels. So they are not going to have saturable binding sites, and they're going to have a sort of quasi-specificity. They'll let some things through and not other things through. But there are porins that will allow pyruvate to cross the outer membrane of the mitochondrion. Now it's in the space between the inner and outer membranes, the intermembrane space, which is going to turn out to be very significant. But there's no porin here in the inner membrane that is going to allow our pyruvate to just diffuse in. What there is instead is an enzyme called pyruvate translocase. Pyruvate translocase is a symporter. Anybody remember what a symporter is? Does what? Things in the same direction, yeah. And so you have one species, pyruvate, that's moving in this direction, another one, protons, that's moving in this direction. Now the protons are going to turn out to be a big deal. For the pyruvate to get across the inner membrane to the matrix, there has to actually be a gradient of protons across this inner membrane. Have to have higher concentration here, lower concentration here. And if that gradient collapses, if the concentration of protons on either side of the membrane is equalized, or if there isn't enough of a difference between the inner membrane space and the cytosol, transport of the pyruvate will stop cold. From that, what might you infer about the mechanism here for transporting pyruvate? Is it likely to be active or passive? Who votes for passive? Who votes for active? The actives have it. That is correct. This is an active mechanism. And the reason it has to be an active mechanism is that the cell is concentrating pyruvate in the matrix of the mitochondrion. That is, yeah, you're generating it out in the cytosol, but then you are moving this stuff into the matrix. So the concentration is actually going to be higher there than it is out in the cytosol. And if you want to move something against its concentration gradient, you've got to spend energy. And the energy here is coming from these protons, which are a source of potential energy. Now, this is a good example of secondary active transport. And we'll get to later how you actually get this proton gradient. But in secondary active transport, the molecule that's doing the transporting is not directly capturing energy from ATP or from light. It's utilizing a pre-existing potential energy source that was created by some other primary active transporter. So now our pyruvate's in the matrix. What's going to happen to it? Well, the first thing that's going to happen to it is we're going to split off a carboxyl group as carbon dioxide. The enzyme that does that is the pyruvate dehydrogenase complex. Whenever you see the word dehydrogenase, you can bet that a redox reaction is involved, and that is the case here. Pyruvate dehydrogenase is a big enzyme complex, and it's kind of cartooned for you here. It's really much bigger and more complicated than it's shown here. But it has three components to it. It has pyruvate dehydrogenase itself, second enzyme called dihydrolipoyl transacetylase, and a third one called dihydrolipoyl dehydrogenase. That's where the redox reaction is going to come in. So the pyruvate dehydrogenase is going to split off the carboxyl group of pyruvate as CO2, the most oxidized form of carbon, leaving behind this 2-carbon unit which becomes coupled to a coenzyme that's attached to the second enzyme here, the dihydrolipoyl transacetylase. And that coenzyme is coenzyme A. And coenzyme A is put together out of some vitamins. Here's pantothenic acid, and here is actually an AMP group. And the pantothenic acid is linked to this short moiety here that's got a sulfhydryl group in its unconjugated form here. And you can release the hydrogen from this sulfur here and couple it to this 2-carbon acetyl group that's left behind when you cut off the carboxyl group from pyruvate. And the bond that's created is a high-energy bond. What that means is if you hydrolyze that, you release a lot of free energy. There is going to be a big negative delta G for doing that. And so we have created a species here that's set up to move this acetyl group from the coenzyme to something else, and that is going to release some free energy for us. So We've now made this species acetyl-CoA, which is bound to the enzyme, and then that is used as a substrate by the dehydrogenase here to generate NADH. That is, we are going to reduce the NAD molecule, we're going to oxidize this guy, and we are now going to capture some energy here in the form of a reduced coenzyme. And this is all happening in the matrix of the mitochondrium. Acetyl-CoA is made from pyruvate, but it can also be made from fats. Fats, remember, are going to consist of glycerol with some fatty acids attached to them in ester linkage. And you can take a fatty acid and you can break it down. And the enzymes that do that break it down 1 2-carbon unit at a time. And so you begin with an enzyme called a lipase that splits the fatty acid off of the triacylglycerol backbone, and then the fatty acid that is released is coupled again to an enzyme that has coenzyme A as its cofactor. And then in repeated cycles of oxidation, you're going to release these 2-carbon acetyl units as acetyl-CoA. And that process is going to require investment of energy in the form of ATP here to produce these fatty acyl-CoAs, but that is then going to get you back a whole lot of reduced electron carriers. That is, as you split off these 2-carbon units, you're making FADH2, you're making NADH. And depending on how long the fatty acid chain is, you get a variable number of these. And so you can generate acetyl-CoA by this different pathway using fats rather than pyruvate as your starting material. You generate reduced coenzyme along the way, which is going to be important later in oxidative phosphorylation. And then you get this 2-carbon intermediate acetyl-CoA that you can feed into the citric acid cycle. So this is your next big step in respiration. You're going to take this 2-carbon activated molecule, acetyl-CoA, and you are going to use it to generate a lot more reduced electron carriers. And once again, just like with glycolysis, I am not interested in you knowing the order of the intermediates, their names, the enzymes that create them, their structures, any of that stuff. I want you to know, what does the citric acid cycle do? What's its job? Its job is to take acetyl-CoA and from it make you a whole bunch of reduced electron carrier. You will also get along the way a GTP molecule if you're an animal cell. And that GTP molecule can be converted to ATP in the cytosol. And so you can say, yeah, you also get an ATP out of the citric acid cycle. That's kind of like a bonus. It's not where most of your ATP is going to come from, but you do get some. But the big picture is that you're making lots and lots of reduced coenzymes here, NADH, FADH2, and you get these 4 reduced coenzymes for every single acetyl-CoA that you put through the citric acid cycle. And how many acetyl-CoAs did you get per glucose molecule? Can anybody do the math in their head? Right, one more. So you get 2 acetyl-CoAs from every glucose, because you got 2 pyruvates and then you get 1 acetyl-CoA for each pyruvate. So for every glucose, you're getting 2 acetyl-CoAs. If you're using fat as fuel, you're getting lots of acetyl-CoAs from that. So in the citric acid cycle, what you do is you take a 4-carbon molecule called oxaloacetate and you transfer the 2-carbon acetyl group from acetyl-CoA to that. And that makes a 6-carbon citrate. And then you go through a bunch of reactions that are isomerizations, dehydrations, but significantly a number of them are redox reactions. Right here in the 3rd step, you generate NADH and you remove a carboxyl group as carbon dioxide again. You do the same in step 4 here. This time you take something called alpha-ketoglutarate and make succinyl-CoA out of it. Then you take advantage of the fact that anything that's conjugated to CoA has got a high energy bond here, and that can be used to drive synthesis of a trinucleotide. Here in an animal cell, it's going to be GDP coupling to an inorganic phosphate to make GTP and regenerating your coenzyme A in the process here. And then you've got a couple of more redox reactions here and here that are going to give you these reduced coenzymes. And you put in this 2-carbon molecule, acetyl-CoA, and during one turn of the citric acid cycle, you got 2 carbon dioxides out. And so a way to think about this is that you completely oxidized 2 carbons that came from glucose. You had already oxidized one of them when you converted pyruvate to acetyl-CoA. Remember, you split off a carbon dioxide there, too. And so in 2 full turns of the citric acid cycle, it's appropriate to say that you have completely oxidized 6 carbons that came from glucose. And you'll hear that a lot. Now, the thing is, they're not all the same carbons from the same glucose molecule. They're just 6 carbons that can be traced back to glucose. But the bottom line is that you are taking a molecule that was at the level of an alcohol, a sugar, and you are ultimately oxidizing those carbons all the way to CO2, the point at which cells can't extract any more energy from the bonds that are in there. That is, you've gotten everything out of it that you're going to get. And this pathway is referred to as a cycle. Now, what makes a biochemical pathway a cycle? Up to now, we've kind of thought about linear pathways when you started with reactant A and then you made B, C, D, E was your end product, and E went off and did something. But what makes a pathway a cycle? Yes? The end product is the starting product. That is, you end up right back where you started, and that's exactly what happens. The last step in the citric acid cycle is you regenerate the 4-carbon oxaloacetate. So big picture, citric acid cycle, you complete the oxidation of carbon from glucose, and you make a bunch of reduced electron carrier. And, oh, by the way, you make an ATP molecule as a bonus. Now, exactly how you do that is a little different depending on what kind of organism you are. If you're an animal cell, you make GTP in your mitochondria. And then the GTP has to be transported out of the mitochondria into the cytosol. And in the cytosol, there's an enzyme that's going to take the phosphate off GTP, give it to ADP, and make ATP. If you're a plant, you've got a different enzyme, and you make ATP right here in the citric acid cycle. And then it just gets transported out of the cytosol and it becomes part of the pool of ATP molecules. So real quick, let's walk through the reactions without bothering to memorize them. Again, we're taking a 2-carbon acetyl group here that's part of acetyl-CoA, and we're attaching it to a 4-carbon molecule, oxaloacetate, to make citrate. We go through this intermediate called cis-aconitate. And we regenerate our coenzyme A in this process, meaning it's free to go back and be coupled by pyruvate dehydrogenase to another pyruvate in a decarboxylation reaction. But we generated now this 6-carbon intermediate citrate, and that is going, oh, this is the cis-aconitate. I'm sorry, I put it in the last one. That's wrong. It's here. The citrate is going to be converted to isocitrate, so that's just a rearrangement reaction here. What we've done is we've moved the hydroxyl group from this carbon down to this carbon. Then we get our first energy yielding reaction in the form of reduced electron carrier, NADH. And so we've got another dehydrogenase here carrying that out, and that is going to convert this hydroxyl group here to a carbonyl group, and the electrons that were withdrawn here are passed to the NAD coenzyme to make NADH. In the process, we remove a carboxyl group here as CO2, and now our 6-carbon molecule has become a 5-carbon molecule. Next step, we make another NADH, another oxidative decarboxylation. So once again, we're going to lop off the carboxyl group here, and we're going to carry out a redox reaction, making a second NADH for us. And in the process, we've coupled coenzyme A here and formed a high-energy bond that's going to set us up for the next step, where we actually release some of that energy in a coupled reaction that allows us to make a nucleotide triphosphate. We take GDP, add an inorganic phosphate, and we've made GTP. And the GTP has to be transported out of the mitochondria and into the cytosol, and there's a very, very abundant enzyme called nucleoside diphosphate kinase, or NDK, and that is the one that will take the phosphate off of any other nucleotide triphosphate, give it to ADP to make ATP. So we just count our GTP as being equivalent to an ATP. Now we're back down to the 4-carbon level. Here, this is succinate. And succinate is going to be the substrate for this particular enzyme, succinate dehydrogenase. And it's an interesting enzyme in the citric acid cycle because all the others are soluble and they're in the matrix. This guy is an integral membrane protein. In fact, it's a very big multi-subunit protein complex, and it's stuck right in the inner membrane. And it is going to carry out an oxidation reaction here. The electrons that are removed from succinate to generate fumarate are passed to the coenzyme FAD to make FADH2. And in fact, succinate dehydrogenase is part of an electron transport system in the mitochondrial inner membrane. And so this is an interface directly between the redox reactions going on in the matrix and then the electron transport going on in the inner membrane. So again, this is the only citric acid cycle enzyme that is part of the inner membrane. The rest of them are all soluble. Then our fumarate gets hydrated to malate, still at the 4-carbon level, and our malate in our final redox reaction is converted to oxaloacetate. So we're right back where we started. But again, the bottom line is for every acetyl-CoA that went through the pathway, we made 3 NADH and we made an FADH2. And oh yeah, we got a GTP equivalent to ATP. So by completing the oxidation of the carbons that were in pyruvate, which of course were originally in glucose, to CO2, which is released as an end product here, we have in fact now gotten all the energy out of that glucose molecule that we can, and we've stored it either in the high energy phosphate bonds of ATP, or we've stored it in high energy electron carriers, NADH and FADH2. Now, I already showed you that we could feed fats into the citric acid cycle by breaking them down directly to acetyl-CoA. The citric acid cycle, in addition to giving us our reduced electron carriers, also gives us a portal for using another kind of fuel, and that's protein. Protein, of course, is a polymer of amino acids, and it's not the body's first choice by any means for use as a fuel molecule, but it can be used as a fuel molecule. And the reason it can is that many of the amino acids that we find in proteins can be enzymatically interconverted with intermediates in the citric acid cycle. For instance, these hydrophobic or nonpolar amino acids can all be converted to succinyl-CoA. Glutamate can be converted to alpha-ketoglutarate. Asparagine and aspartate can be converted to oxaloacetate. And so if you're kind of living on a barren island and you run out of your Twix bars and the other stuff that you brought along, and then you eat all the bananas and the coconuts and everything else you can find, and then your body says, well, we're out of carbohydrate, what do? Oh yeah, we can burn fat, and then we can make acetyl-CoA from that, and our citric acid cycle can give us ATP. So we're still good, but then eventually you burn all your fat left. What comes next? Protein. And where's most of the protein come from? Your muscles. And that's when you begin to wither away. That's when you become the skinny little man or woman who's crawling along on the beach going, save me. Because if somebody doesn't come along soon with some carbohydrate or some fat, your body's going to run out of protein to burn via the citric acid cycle, and then you're going to reach equilibrium because you can't make any ATP anymore. So the citric acid cycle is a central player in catabolism because not only does it give us most of the reduced electron carrier that we're going to use to make ATP and oxidative phosphorylation, but it also gives us a portal for using alternative fuels. We don't have to have glucose. We don't have to have fat. We can get by with protein, but you don't get as much bang for your buck when you do that because you've already bypassed many of the ATP yielding steps when you do that, and you have to spend energy to get those amino acids into the form of the intermediates of the TCA cycle. So here are the things to know about pyruvate oxidation and the citric acid cycle. How does the pyruvate get into mitochondria? Form of active transport, secondary active transport using a symport mechanism in the inner membrane. What part of the mitochondrion is involved in metabolizing pyruvate? It's the matrix, the soluble part. What two products do you split pyruvate into? 2-carbon acetyl-CoA, 1-carbon CO2. And the CO2, of course, diffuses out of the mitochondrion, can actually diffuse out of the cell altogether, released as a waste product because it's got no more energy that we can use. One of those products enters the citric acid cycle. I already gave it away. It's not the CO2, it's the acetyl group, the acetyl-CoA. It is a cyclic pathway, the citric acid cycle, because you start and end up at exactly the same place, oxaloacetate. What does the citric acid cycle make? It makes reduced electron carriers. For every acetyl-CoA, you make 3 NADH, 1 FADH2, and you get 1 ATP out of it as a bonus. What happens to the GTP that's made in mitochondria in animal cells? It gets transported out to the cytosol where that enzyme NDK takes its phosphate and gives it to ADP to make ATP. All of the enzymes in the citric acid cycle are soluble except for one. Anybody remembers which was the one? It's a redox enzyme, and so what did I say a lot of them were called? Dehydrogenase or dehydrogenase. So yes, succinate dehydrogenase. You guys should remember that one. That's the one that's membrane associated. How many turns of the citric acid cycle are fully needed to oxidize 6 carbons that came from glucose? 2, correct. But remember, it's not from the same glucose molecules. And by the way, when I was an undergrad, I used to have this picture in my head of little circles of enzymes floating around the mitochondrial matrix, and that's how the citric acid cycle worked. It doesn't work that way. These enzymes that are soluble, they're diffusing all around. So are all their substrates. It's a pool of enzymes, a pool of intermediates, a pool of the enzymatic substrates here. The only thing that's kind of fixed in space is the succinate dehydrogenase complex, but there's lots of them. And so these things can go anywhere they can be bound. And so it's not, as simple as saying, yeah, we took this carbon and then, that carbon that was left behind stayed out there and waited until that cycle completed and then the next one came around and so forth. Not that easy to track it, okay? However, in 2 complete turns of the citric acid cycle, you completed the oxidation of the remaining 4 carbons that came from glucose. Remember that the first 2 you got rid of from pyruvate in that step where you made acetyl-CoA. How do you use fats as fuel? You break them down to acetyl-CoA. How do you use amino acids from protein? You turn them into intermediates of the citric acid cycle. Let's see where we're at. We're at stopping time. So you guys have a great weekend. Let me know if you've got any questions. We'll finish this up on Tuesday. Yep. Mm-hmm.
Is that when your body goes into like ketoacidosis?
Okay, the thing about ketoacidosis is
admit I don't know